Our topic is molecular techniques in isolation of genetic material and gel electrophoresis in principle and process of biotechnology screening for recombinants, Unit 8, Chapter 4 in botany. So, what is the molecular techniques used in the isolation of genetic material and how the gel electrophoresis is helpful in this technique? So, electrophoresis is a separating technique used to separate different biomolecules with positive and negative charges. So, the electrophoresis, that is the electrophoresis, is used to separate the different molecules using the current with positive and negative charges. So the principle of it is by applying the electricity, that is DC current, the molecules migrate according to the type of charges they have. So when the current was applied, the molecules will move according to the charges. That is, negative means it moves towards the positive charge and positive means it moves towards the negative charge. Like that they will move. The electric charges on different molecules are variable. So the charges will be vary. So as I said, the positive charge cation moves towards negative charge cathode and the negative charge anions moves towards positive charge anode. Like this the movement will be, that is the molecules will be attract. Okay, the opposite signs will attract, no, that was done here. Agarose gel electrophoresis. It is used mainly for the purification of specific DNA fragments. So only few specific DNA fragments can be purified with this gel electrophoresis. Agarose is the convenient for separating the DNA fragments ranging from the size few hundreds to about 20,000 base pairs. So the Garros gel electrophoresis, the Garros is convenient to separate the DNA size ranges from few hundreds to 20,000 base pair. This they may ask in your 1 mark as well as in your NEET exam. Polyacrylamide is preferred for the purification of smaller DNA fragments. So smaller DNA fragments can be purified with the help of polyacrylamide. So they can ask this also in your 1 mark. Okay, polyacrylamide. So the gel is a complex network of polymeric molecules. So the gel is a complex network made up of polymeric. Poly means many substances are involved in this gel. The DNA molecule is negatively charged molecule. Under an electric field, the DNA molecules migrate through the gel. So, as we have discussed about the DNA is a negative charge. In the previous classes we have discussed, no? So, due to the negative charge, that when the current was applied, the DNA migrate along with the gel. Okay. So, the electrophoresis is frequently performed with marker DNA fragments of known size which allow accurate size determination of an unknown DNA molecule by interpolation. So, interpolation means what? Means it will change. So, this gel electrophoresis has marked DNA size. With the help of the marked DNA size, already you have it, you had in this plate. With that, the unknown DNA size will be notified. Okay, so the changing of the size can be seen here. The advantage of a Garros gel electrophoresis or that the DNA bands can be readily detected at high sensitive. So the advantage in this gel electrophoresis agarose, the DNA bands are readily detected at high. So you can see these bands, no, they are readily detected. Okay, because they are highly sensitive. The bands of DNA in the gel are stained with a dye called ethidium bromide. This they may ask in your 1 and as 1 mark as well as in the NEET as well as in the board exam. So the staining of the staining used in the gel electrophoresis to form the bands of DNA. That dye is ethidium bromide and the DNA can be detected as possible fluorescent illuminated in UV light. So we can see through the UV light. You can see means you will get like this. In the fluorescent UV light you can see these bands which will give orange fluorescence. So the color will be, the color of the DNA will be in orange fluorescent color, that is orange color. So which can be photographed. So while getting the orange color, fluorescent color, you can take a photograph. That is, the photograph was taken like this is the one when the gel electrophoresis was conducted and it was taken the photograph. So this is a photograph and this is the instrument of gel electrophoresis. Okay, hope you understood. If you have any doubts, post your doubts in the comment box. Give a thumbs up, share and subscribe to Science Easy Tech channel.